Good morning, everybody, and thank you for the opportunity to be here. I would like to talk about environmental sampling and sample preparation in connection with biosafety. But before I start, I'd like to mention that my background is in environmental research, not in traditional biosafety. And the aim of this presentation is not to give definitive answers, rather to raise questions about this field. So about environmental samples, when we think about environmental samples, I think we should consider two aspects in connection with biosafety. First, that there is or there could be some level of biosafety risk in connection with every sample or every sampling event if it's not done properly. So if the national or international regulation standards or good practices are not followed. For example, one can get a skin infection during sampling a poorly maintained swimming pool, or even sadly in the past there were deathly infections because of sampling a wastewater plant without the proper protective equipment and by an immunocompromised person. So we always should consider the biosafety perspectives also. And second, it's not typical, but not excludable that environmental samples can also represent high-level biosafety risks. And what am I talking about? I have collected some samples. First of all, let us think about drinking water, for example. Drinking water is very vulnerable because if intentionally contaminated, the contamination can affect a very large population, and it can lead to a severe public health threat. So it is not a risk that we can completely exclude. And in the case of anthrax, even soil or water is not the primary source of infection. It can be a source, and in several cases, analyzing of these environmental samples can be necessary. Or even wastewater from biosafety facilities can be a sample of high risk level, but with the properties of an environmental sample. And in the fourth blue box, I have collected some samples of, let us say, unknown risk levels. For example, landfill sites can also, not generally, but can pose a high risk level. And why is it important, or what is different with these samples? Maybe it can pose a dual challenge, because environmental laboratories are prepared for these environmental samples, for general environmental samples. We have the knowledge, the proficiency, and the equipment designed for the sampling and the sample preparation for environmental samples. However, usually environmental laboratories are not qualified to handle samples above BSL-2 level. And on the contrary, biosafety laboratories do know how to handle these very high-level risk samples. However, they may not be familiar with the special requirements for environmental samples. They may not have the experience or the equipment required for this task. of course, I'm generalizing, and it is not true, or may not be true for all of the laboratories, but there may be some gaps of knowledge or equipment on both sides. So maybe knowledge sharing is a good idea on this field, or even we could develop some common good practices. And a few words about environmental sampling and sample preparation from the environmental research side. As I think for all samples, sampling is a very important step which determines the adequacy of the whole test. It is especially important in case of environmental sample to adequately choose the sampling points and the equipment to have a representative sample. And usually these samples are of large volume, even up to several liters. And preservation also may be needed in terms of microbial samples, maybe just refrigeration, but also preservation can be necessary. And if there is a biohazard risk, the sampling staff should manage these special risks throughout the entire process. As I have mentioned these examples, the appropriate protective equipment or the attention of the vulnerable staff members can be vital in these cases. And in general, how do we do sample preparation of environmental samples? First, usually there is a homogenization step, and then in the most cases, the water samples can be processed directly, while soil samples requires pre-treatment steps. During these steps, we must focus also on the biosafety issues. Maybe there is an opportunity for sterilizing steps or other steps that can decrease the risk and ease the work. But we have to think about it also during the sample preparation. In terms of environmental samples, the main goal of sample preparation is usually concentration, because analytes are present in low concentration. And in several cases, the matrices that we have to get rid of are very complex. So we may need some sample cleanup as well, or highly selective methods. In samples collected for microbial analyzing, they are usually have to be processed within a day, and usually a subsample is tested after homogenization. And concentration is done by filtration. In the case of bacterial organisms, it's a simple membrane filtration. However, for smaller viral organisms, which represent a lower size range, we have to use lower pore diameter filtration, for example, ultrafiltration, or some special methods like size increase with flocculation. And then we can do the analysis by culturing on media or by extraction of DNA or RNA. In the case of chemical contaminants, the time window is usually wider. We can use preliminary rapid tests, but maybe the most used method is, or after these tests, is solid phase extraction. With this technique, we can concentrate and clean up our sample, and at the end of the process, we have a small volume concentrated sample ready for measurement by immunological, mass spectrometry-based, or functional assay-based methods. So to process our samples, we do need some special equipment and space and physical so we have some difficulties with this maybe, because for example, for filtration or the solid phase extraction, we have to provide under or over pressurization of the system, and we have to have these equipments to carry out the sample preparation. And we have to think about this process and have to define the level of safety which is necessary for the sample preparation of a sample. And to summarize my really short question-raising presentation, I think it is very important to ensure biosafety for all type of samples, including environmental samples, and knowledge sharing could be useful and important in this area also. And maybe however, it's not very typical or not very often. We should think about that, how to handle these high risk level environmental samples, and maybe do the high level, high biosafety laboratories have to be prepared for these samples or not. And thank you for your attention.